So today I'm going to be talking about best practices in recovery from blood draws to tissue staining. And just before I start, I do want to give a warning. So there are a few graphic images and videos. So if you're not comfortable with them, feel free to avert your eyes. But I tried to lessen them as much as I could, but these are very invasive. So just keep in mind. So I'm going to be talking about three different aspects of rodent biomethodology: blood draws, injections, and histology. So I believe that these are very foundational aspects for any rodent experiment because usually you'll inject, you'll see some effects through blood draws, and you'll take a look at histology and even histopathology to see, are my injections causing cell death or toxicity or what's going on? So I'm going to be going into all three of these. So to begin, I'm going to be starting off with blood draws. So our lab in particular uses blood draws for a variety of reasons, most notably ELISAs and cytokine assays. So I'm going to be going over 5 very common types: mandibular lateral saphenous vein, retroorbital venous sinus, intracardiac, and tail vein. And so we'll start off with mandibular. So mandibular blood draws are the type that our lab actually uses almost 100% of the time. And they're actually one of the most simplest forms of blood draws that you can do, which I do believe is why our lab also uses them. And so for the materials, you're going to need a collection tube, which we often use Eppendorf tubes, gauze pads, 5mm animal lancets, and alcohol wipes. So for the general procedure, this is probably as simple as it'll be. And so you'll first prep the area that's going to be punctured with the alcohol wipes to just stop it from disinfection. And so once you puncture, the blood's going to be seeping out. You just need to make sure that that area is clean. And so you'll then restrain the rodent. So there's multiple types of ways to restrain the rodent. You can use double hands and manual restraint or single-handed. And I won't be going over that in this PowerPoint, but if you'd like me to explain that, I could. And so then you have to locate the vein. So there are a few markers on the mouse that can help you locate the vein. The vein that we're aiming for is this facial vein right here, as in labeled in the picture. And so there's a few markers that can help you. So there's a dot on the mouse's cheek that is referred to as the dimple. You can either go just directly above that, and what I do is I go directly above and little to the left, just keeping in mind where exactly the jaw curves upwards. And so once you do locate the vein, you can go ahead and take your lancet and puncture perpendicularly to that vein. And if you're lucky enough to have hit it, then you can then collect the blood. After that, you do take a gauze pad and stop the bleed by holding it to the cheek for about 20 seconds, or at least until the blood stops. So with this blood draw, if you go too far back on the vein, you'll hit the external jugular, which will cause bleeding to come out of the ear and the nose. In that case, you can still stop the bleed just from where you originally punctured, but you do have to inject the mouse with 0.9% saline to help its blood cells proliferate again. And so now onto the lateral saphenous vein. So I actually haven't done the lateral saphenous vein puncture before, and it is a little more complex than the mandibular form. So for this is one of the few that the mice do have to be anesthetized through an anesthesia chamber, and you do actually have to shave the area of the mouse with electric clippers. So you then need gauze pads, a petroleum-based lubricant, alcohol wipes, a 22-gauge hypodermic needle, and a hematocrit tube blood collector. So for this procedure, again, you have to anesthetize the rodent in an anesthesia chamber, just general anesthesia, no local. You then have to prep the area with the alcohol wipes, and you have to clip the hair from the lateral lower leg. So the reason you have to clip the hair from the lateral lower leg is because this vein is very superficial. So the only way to really accurately see it is by clipping that hair. So after you clip the hair, you then apply a thin layer of petroleum-based lubricant, which is going to help the blood bead a little better. And so it'll be really helpful for like actually collecting it so it doesn't come out like in a non-beadly fashion. And so after that, you do constrict the vein using your thumb and your index finger. And again, you take your 22-gauge hypodermic needle, puncture it, and you can then collect the blood. And once again, you take your gauze pads and hold it to the area for at least 20 seconds or until the bleed stops. So I do have a video of the saphenous vein bleeding. It is a little graphic, so you can go ahead and avert your eyes if you do get uncomfortable during this. It is a really quick video. So as you can see, they're currently clipping the hair from the leg, applying the petroleum-based lubricant. And so they're kind of just rubbing that in to help for easier blood collection. And now they're taking their 22-gauge hypodermic needle and they're just going to stick it in to get into the vein. And the vein is very superficial and you can actually see it in the video. And now the blood is coming out, and because of the petroleum-based lubricant, it is very in like a bead-like fashion. You can go ahead and take it up. So retroorbital venous sinus. So the retroorbital venous sinus blood draw is actually a blood draw from the eye. So contrary to popular belief, it's actually not directly in the eye, but it's in the sinuses around the eye. And so for this one, you're going to also need local anesthesia along with general anesthesia. So again, your hematocrit tube blood collector, gauze pads, and a sterile eye ointment. So I personally believe retroorbital venous sinus punctures are actually the simplest form of blood draws. After I did all my research, I do believe that this is actually the simplest version, but it is a little more graphic, so a lot of people do opt out of it. So for the procedure, you anesthetize the rodent generally in an anesthesia chamber, and then you can either take proparacaine or tetracaine to do local anesthesia on the actual eye. And so afterwards, you insert the hematocrit tube into the sinus just right around the eye, and you rotate the tube until blood is going to flow. So you're going to feel a pop, and that is the vein popping, and that's when blood is going to flow into your hematocrit tube blood collector. Once you stop the bleed, or once you take all the blood that you need, you can go ahead and insert sterile eye ointment, which is kind of the equivalent of rubbing it with alcohol wipes. And you can then hold the eyelids closed with a gauze pad to stop any residual bleeding. So I also have a picture of orbital bleeding, and this one actually is a little more graphic, just warning. But it is a very short video. So this is actually a rat, and they're currently, you can see they're sticking the tube collector into the eyelid. And you can see them twist to kind of pop the vein. And so once they actually do pop it, let's see, you can see that blood starts flowing out, and they go ahead and collect it. And so after this, they do just stop the bleed with a gauze pad after inserting their sterile eye ointment. So now on to intracardiac punctures. So intracardiac punctures are a little different. These are actually a terminal procedure. It's when you're kind of just done using the animal for your experiment. You want one good big blood draw from the heart, and you're going to euthanize it at the end. So for this, you're going to need anesthesia because the mice do have to be in deep anesthesia to do this, a syringe, a hypodermic needle, and alcohol wipes. So as before, you anesthetize the rodent just with general anesthesia, but it has to be very deep anesthesia because they are going to be euthanized at the end. And you're also going to be using a mask to keep them under while you're actually doing the blood draw. You're then going to prep the area by using alcohol wipes, and you're going to insert the needle after you find where the heart's beating. You're going to insert the needle right under it in its abdominal cavity, and you're going to aspirate the syringe to just take the blood out. Afterwards, you can use any method of euthanasia to just put the mouse down. So now onto tail vein. So tail vein punctures are actually one of the more difficult methods of blood draws. They actually take the longest, and they're the hardest to do, in my opinion. So for the materials, these are a little different. You're going to need a syringe, a hypodermic needle, gauze pads, a plexiglass restraint box, a warming source, and alcohol wipes. So first, you're going to be prepping the tail with alcohol wipes before it's punctured, and you have to restrain the rodent. But it's not the same restraint that's used for any other method. In this case, you do put it in a plexiglass restraint box, and you kind of squeeze it in there so that the mouse can't move, but its tail is still taut. So once you do restrain the rodent, you also have to make sure the mouse is still breathing because it is very hard to tell once it is squished in that box. You can then use a warming source to locate the vein. The warming source allows the vein to kind of pop out a bit so that it's easier to find it in the when you're actually doing the blood draw. And so if you're lucky enough to find it, you can go ahead and puncture it with the hypodermic needle and aspirate with the syringe to collect the blood. Afterwards, you can just hold a gauze pad to the area and stop the bleed. So I do have a video of this one as well. And so I'm going to be skipping forward a bit. But they don't actually show the warming source, but you can see they're putting the mouse in the plexiglass constraint. They kind of squeezed it in. And now they're going to find the tail vein. And so you can see they go get in there and they just pull the blood out, like as you can see right there. So now I want to go into an investigation into the effects of different blood collection methods on indicators of welfare in mice. So the welfare in mice is important not only because it is for their welfare, but also for our experiments. So when mice are stressed, they tend to secrete things into their blood, most notably corticosteroid, which I will go into, and hemoglobulin. So while these don't directly affect what we do, there's still a confounding variable that could affect our experiments in one way or another. So to begin, they used 72 mice of this strain with 12 mice in each group, and they tested 6 methods of blood collection. Retrobulbar bleeding, or RBB, and they tested RBBA, which was a thick filament, and RBBB, which is a thin filament. They then tested tail vein bleeding, TVB, saphenous vein bleeding, SVB, and facial vein bleeding, FVB. And finally, jugular vein bleeding, which I didn't go over, but is very similar to the mandibular blood draw or facial vein, and JVB. So they looked at the corticosterone concentration. So they found that plasma corticosterone is actually secreted in the mice's blood when they're stressed. It comes from their hypothalamic pituitary adrenal gland, and it's elevated in mice that are very stressed, and it can actually affect some experiments where they're looking for glucocorticoids. But for our experiments, not so much, but it's still a confounding variable. Found is that retrobulbar bleeding, the one in the eye, is actually much less stressful for mice than any other type. Facial vein and jugular vein actually secrete the most corticosteroid concentration, and keep in mind that our lab actually does facial vein bleeding more commonly than others. And so they then looked at in-cage activity. So in-cage activity is after they've drawn the blood, how do the mice act in their cages? They rated them on a scale of 1 to 7, with 1 being immobile, 4 being visible interest in the environment, and 7 being active with strong exploratory and locomotor behavior. So RBBA actually got a 7 score, tail vein bleeding also a 7, RBB just below, but once again, facial vein bleeding and jugular vein bleeding were actually the lowest score of activity. And facial vein bleeding and jugular vein bleeding, we can see that they also had the lowest score in activity, which means that they are more stressed because less stressed mice will move around more after blood draws, which also explains why the facial vein and jugular vein had more corticosteroid concentration released into their blood. And so they also looked at bleeding length. So typically, the longer it takes to bleed the mice, the more stressed they're going to be. And so you can see with retrobulbar bleeding and retrobulbar bleeding A and RBBB, these took the least amount of time, followed by facial vein, and tail vein actually took the longest time. And so as I've said before, tail vein, you do need a warming source, a restraint box, you have to locate the vein. So it makes sense that it is the longest. But as you can see, once again, retrobulbar bleeding is at the lowest time, the lowest stress of in-cage activity, and the lowest corticosteroid concentration. They then looked at an open field test. This open field test has 4 different quadrants, 2 of which are covered up in dark, while the other 2 are in open fields. So if the mice are expressing an exploratory behavior, they will go into the darker spaces. And the mice that do express this exploratory behavior are typically the ones that are not as stressed because if you're anxious, you're not going to be going into those dark areas. So as we can see, their latency to move is low for most of them, except for jugular vein bleeding, a little higher than most facial vein, but not really. But when you look at distance traveled, they all do vary. But once again, for RBBA and tail vein bleeding, they are still pretty high up there compared to facial vein and jugular vein. So facial vein and jugular vein so far has shown a trend of mice being more stressed while they are being bled. And so once again, keep in mind that our lab does facial vein bleeding most of all. So the authors also went into the advantages and disadvantages of each bleeding method. And so I've already kind of covered most of these, but personally what I do feel is best is retrobulbar bleeding. It's the least stressful, it's the fastest, but a lot of people are adverse to use it because it is sticking a needle into the eye. But I do believe that you can get a large volume of blood from it, and it's typically not detrimental if you are experienced. But if you are not experienced, you can actually lead to a lot of side effects, but that's for kind of all of the blood draws. So now I want to go into injections. So I'm going to be talking about 5 different types of injections: subcutaneous, intraperitoneal, intradermal, intramuscular, and intravenous. And so our lab uses injections for a variety of things, injecting different substances, different amounts of substances. I do believe that they are very fundamental to most mice experiments. So I first want to start off with subcutaneous. So subcutaneous injections our lab does probably most often. Not that we need to, there are many different types of injections, but it is very simple and easy, and you can inject a high amount. So it's an injection over the loose skin on intrascapular, which is in the picture on the right, or the hind leg area, which is on the picture on the left. So you do have a 2 to 3 ml maximum that you can inject, and you have to inject at a 30-degree angle. So when you're injecting, you can go ahead and aspirate the syringe a bit to see if you get any negative pressure. And usually if you do get negative pressure, it's an indication that you're in the right area, but it's not confirmation. But when you do inject, you should see a pocket form on the hind leg of the mouse, and that'll kind of be confirmation to you did inject in the subcutaneous area. So I do have a video of the subcutaneous injection. Once again, it's a bit graphic, so feel free to avert your eyes. So they're in the hind leg area. They're currently injecting. They don't aspirate the syringe, but when they do inject just now, you can see that there's a pocket forming on the body of the mouse. And so that is an indication that you are in the subcutaneous area and you did the injection correctly. And so now on to intraperitoneal. So intraperitoneal injections are injections directly into the body cavity. They're generally done on the inguinal area of mice, and you can only inject a maximum of less than 3 mils. And in order to make sure that you are in the correct area, you should do it at a 40-degree angle. And inguinal area at a 40-degree angle, chances are that you are into a body cavity. So intraperitoneal injections are actually not one of the more complicated injections that you can do on mice. And intraperitoneal injections are actually mostly used for drugs being injected into the body, but they can also be used just as another way to do tail vein injections. If the tail vein injections don't work, you can just go to intraperitoneal. So for intradermal. So intradermal injections are injections directly into the dermis of the skin. You can only inject about 0.05 to 1.1 mil, and they must be done under anesthesia. So these are actually one of the only types of injections that can be done under anesthesia. And so that is because you have to keep the skin taut, and with one hand, you can't actually do that. So as you can see in these pictures, they do kind of pinch the skin upwards just to make sure that they are in the correct dermal area and not above or below. So now for intramuscular. So intramuscular injections are injections directly into the caudal hind leg muscles, the cranial thigh muscles, and you can only inject a maximum of 0.05 mils. And this is commonly used for injectable anesthesia. If you don't want to put your mice in a chamber in order to be anesthetized, you can use an intramuscular injection in order to anesthetize your rodents with injectables. And as you can see in this picture, this is the hind leg muscle that they are injecting it into. And now onto intravenous. So I'm going to be talking about two different types of intravenous injections, tail vein injections and the retrobulbar injections. So tail vein injections are actually very similar to the blood draws for tail veins, but instead of aspirating, you're going to be injecting. So this is an injection into the lateral tail vein once again. You are going to put it in a plexiglass restraint box, as you can see in the picture on the right, and you're going to dilate the veins using a warming source. And you can only inject 0.2 mils maximum. As you can see on the right, so they found the vein and they're currently injecting it. And the other tail vein injection, or the not tail vein injection, venous injection, the retroorbital venous sinus. So this is an injection directly into the orbital sinus. It's very similar to the blood draws, but except instead of twisting your hematocrit tube, you're just going to be injecting something in that same space. So the volume of the liquid that can be injected is 0.15 mils, and it is an alternative to tail vein injections. While you can't inject as much, it's much faster than tail vein injections, and you can probably do less damage to that area if you do retroorbital venous sinus, but it's more of a choice of personal preference. So now I want to go into tissue staining. So tissue staining is a very fundamental aspect of rodents because in order to understand truly what's going on inside of the body, you need to look at the morphology of the tissues to know, are there any defects or abnormalities that are occurring because of the research that you're doing? So I'm going to be going over 3 different types of tissue staining: the tunnel assay, hemotoxin and eosin staining, otherwise known as H&E, and immunohistochemistry, both direct and indirect. So I'm first going to be giving a general overview of each of these 3 topics. So the tunnel assay. So the tunnel assay is a method used to visualize and quantify cells that have undergone programmed cell death, otherwise known as apoptosis. So this can be used in many experiments in labs, such as to see if injections are causing cell death, or even to test the efficacy of new treatments for disorders that are characterized by the inhibition of apoptosis, such as cancer. And so while I will be going over this in more detail soon, the basis of the tunnel assay is that when cells undergo apoptosis, they leave behind DNA fragments that have nicked ends that have 3' hydroxide groups coming off of it that can be attached to. And so for H&E staining, H&E staining uses 2 solutions, hemotoxin and eosin. So eosin is a pink red stain and hemotoxin is a purple blue stain, and each of them have specific areas that they can only stain based on acidity or basicity, and I will also be going over this in a bit more detail in a few slides. So now for immunohistochemistry. So immunohistochemistry is used to detect specific antigens using antibodies, either only a primary antibody or also a primary antibody and a secondary antibody, depending on if you're doing the direct or indirect method. And this is basically used for if you want to see protein localization, life cell processes, or cell or tissue morphology. And so all 3 of these types of tissue staining follow a very similar general procedure that I'll be going over together. So first off, tissue fixation. So tissue fixation is very important to stop all the biochemical reactions that are occurring in your tissues in order to preserve the antigen, keep them from decay and putrefaction. And so to fix the tissues, after you sacrifice the mouse and harvest its organs, you can go ahead and wash it in 0.9% saline and then transfer it to 4% paraformaldehyde for 4 to 24 hours at 4 degrees Celsius. And so next up, after the 4 to 24-hour period is over, you can go ahead and take your tissue and place it in 70% ethanol to get it ready to be dehydrated. Tissue sectioning. So our lab uses paraffin embedding, but cryosectioning is also a very popular method of tissue sectioning. So cryosectioning involves first freezing the tissue in a mixture of isopentane and dry ice. And so once your tissue is frozen, you can then use cryosectioning, which is the picture on the left, in order to slice it in sizes about 4 to 10 microns. But although cryosectioning is a very popular method, you have to be experienced to use it because there's a lot of things that can go wrong. So if your tissue doesn't freeze as fast as you want it to, so crystal formation can actually occur under the tissue morphology, creating what they call a Swiss cheese effect when you look at it under a microscope. So while paraffin embedding does actually take longer, it is more accurate, and that's the one that our lab actually uses the most. So for paraffin embedding, your tissues first have to be dehydrated. So as I said, after the 4 to 24-hour period of tissue fixation, you can go ahead and put it in 70% ethanol, and from there, higher percentages of ethanol until xylene. So once your tissue is dehydrated, you can go ahead and use a tissue processor to put it in a block of paraffin wax, which you can see in the picture in the middle, which is actually taken in our lab of about 2018 when I was actually learning to embed organs in paraffin. And so from paraffin embedding, you can go ahead and slice the tissues in about 4 to 10 microns in size using the tissue processor. And so after you dehydrate the tissues, you then have to rehydrate them again. So this step is referred to as permeabilization, and you're going from nonpolar to more polar reagents. So as I said, first to dehydrate it, you were going from about 70% ethanol to 100 to xylene. So now you're doing the opposite. You're going from xylene to 100% to 90, 70, 50, and then 30% ethanol. And so you can do this manually as seen in the photos below, but there's actually a machine in BSB that does it for you. And so the basis of this is that if you have to rehydrate it in order for the tissue to be able to penetrate into the cell nucleus in order to actually stain the tissue. And so now let's go on to actually stain the tissues. So as I said before, so the tunnel assay is based on how after cells go through apoptosis, there are DNA fragments that are left behind with nicked ends that have free hydroxide groups. To these hydroxide groups, fluorescently labeled DUTP attaches on it with the help of TDT enzyme, which is a catalyst that allows it to actually be connected with the cofactor of the labeling buffer with cobalt. And so once this is added to the tissue sections, you can go ahead and incubate for 1 to 3 hours and use stop buffer after that period of time in order to stop the tunnel reaction. Once that is done, you can then use fluorescence microscopy to look at your tissues under the microscope. So for H&E staining, hemotoxylin is a basic dye that's going to stain acidic structures. So it stains the nucleus and any other structures such as DNA or RNA and ribosomes from the rough endoplasmic reticulum. And so that's why you see the purplish blue stain of the nucleus in all of those H&E stains. And so for eosin solution, excuse me, eosin solution is an acidic dye that's going to stain basic structures such as the cytoplasm. It's actually one of the most popular dyes to stain the cytoplasm, but it also stains because most proteins in the cytoplasm are basic. And so it also stains like cytoplasmic filaments of muscle cells and intracellular filaments. And so they just stain them pink and red. So when you see an H&E stain, the blue and purple color are actually the nucleus, the DNA, the RNA, and the pink red stain is the cytoplasm and any other basic proteins. And so now for immunohistochemistry. So to stain cells or tissues using immunohistochemistry, there's two methods, the direct and indirect. So the direct method, you use a labeled primary antibody that's attached to the antigen and only that. And so the direct method with the labeled primary antibody, this is actually less sensitive because you don't have as much binding as the indirect method. So most people don't opt out of using this because it's also more expensive to buy a labeled primary antibody versus using a secondary antibody that you can use with multiple primary antibodies. And so for the indirect method, you do have an unlabeled primary antibody versus the direct method's labeled primary antibody. And you can use a labeled secondary antibody to connect to that that's going to fluoresce. And you can label that with FITC, rhodamine, or Texas red, and it's going to be a lot more sensitive because there is more binding going on, and you can go ahead and look at your tissues under the microscope. And once again, it's going to show you live tissue processes, cell morphology, and the like. And so I want to go over an investigation into the histopathology of the liver, kidney, spleen, and eyes exposed to gold nanoparticles. And so I'm going to be more focusing on the histopathology aspect, not so much on the gold nanoparticles aspect. So what they were basically trying to do is see what, so they haven't looked at the toxicity of gold nanoparticles. And so in this experiment, they were looking at what does different dosages of gold nanoparticles do to the liver, kidney, and the spleen in mice. And so they took 60 adult Swiss albino mice with 6 mice in each group. They had a control, 6 groups treated with a single intraperitoneal injection of 5 nanometer gold nanoparticles, 20 nanometer, and 50 nanometer. They then took 3 groups that received intraperitoneal injections twice on days 1 and 7 of 5 nanometer gold nanoparticles, 20 nanometer, and 50 nanometer again. And so starting off with the liver, so we're going to be looking at some of the diseases that were caused by these higher injections. And so you can see image number 2. So image number 1 is a control of how a healthy liver looks. And so image number 2 is, we can see that these white blobs, that is actually steatosis. It's the accumulation of fat in the liver. And how this happens is that toxins elevate liver enzymes, which lead to the augmentation of fat. And so if you do see hepatosteatosis in your tissue sections, it does mean that there is something going on that's elevating your liver enzymes, most notably toxins from higher percentage of injections. And so you can see the arrows are pointing to these white blobs, and that is actually fat. And so now we're still looking at the liver, and we can see, we're still looking at the liver, and what you can see, what I've circled, this is actually the aggregation of inflammatory cells that were called to the site because of something abnormal going on. And so most notably, the toxicity from the higher injections. And you can see right here with the arrows pointing to, this is actually degradation of the cytoplasm, which occurs when cell death occurs, which from what I'm assuming in the paper was because of the higher dosage of the gold nanoparticles versus the lower dosage that they've used in the past.
Sarah, I have
So
a
you can
question
see,
for you.
yes.
What is the scale, the size scale for these images? Like, what would be a good way of saying the magnification used on the microscope? I think we use very low magnification when we do our imaging, and I'm wondering if that's inappropriate.
If what's inappropriate, the magnification used on this
the
PowerPoint,
magnification we use.
yeah.
I don't know if when we're looking, I don't know if we'd be able to see these little white fat globules that, or maybe we would. I'm not sure. At the magnification we use.
I don't exactly have it on my PowerPoint, but I do have the paper saved that I can look for it and tell you about that after the PowerPoint. But I don't have it on me right now. Yeah, but I do have the paper saved, so I can go ahead and pull it up afterwards. And so what I was saying on this slide, so this white area right here, that is cytoplasmic degradation. And so you can see that that is a blank area. It can be confused with necrotic foci, which I will show in a few more slides, but this is actually just degradation of the cytoplasm. So in here on picture number 6, you can see that this blob right here in the corner, that is an activation of a Kupffer cell. So Kupffer cell is actually a resident macrophage in the liver that comes to sites of injections and release cytokines. So generally, if you do see Kupffer cells in your liver, it's not a good sign. It basically means that there is something going on in your liver that is not healthy, that there are toxins. And so I do believe that again is a cause because of the high injections of gold nanoparticles that they did on these mice. And so once again, these are Kupffer cells, and they can actually activate or call macrophages to the scene of the inflammation or infection. And so here, this is a very distorted picture of the liver. I believe this was for a 50 nanometer gold nanoparticle injection. And so you can see there's multiple necrotic foci, which are these white spaces, not to be confused with the degradation of the cytoplasm. But you can also see there are infiltrative cells, these blobs right here, which can be confused with inflammatory cells, but these are actually just infiltrative cells that come into a site of infection to just kind of check what's going on. And so you can see that there are multiple necrotic foci around here. I would have circled it, but the whole thing is kind of just messed up. But this does also happen because of elevated toxins in the body. So this is again the liver, and you can see there is multiple aggregation of inflammatory cells, and in the middle is edema. So edema is when it fills with fluid, and this only really happens when there is an infection or elevated amounts of toxins in the liver, like specifically. And so once again, I do believe this is because of the higher injections of the gold nanoparticles that occurred in the mice. And so you can see this pink area is fluid filled, and it is surrounded by inflammatory cells. Oops. And so now onto the kidney. This picture is a picture of distorted glomeruli in the kidney. Usually, only the glomeruli get distorted if there is elevated amounts of toxins in the body or if there's something going on in the kidney, such as an infection. A kidney infection can lead to distorted glomeruli, and you can see there is multiple areas where the glomeruli is actually eroded. These white here. But as you can see in comparison to this picture, there are a lot of inflammatory cells and aggregation of them in certain areas. And so this is something key to watch out for if you are looking for toxicity in your kidney. So now to this picture. So I actually pointed to arrows this time. So these are actually giant macrophages that, or sorry, this is the spleen. So these are actually giant macrophages that are in the spleen. And you can see that part of the spleen has actually gone away and it's actually filled with these pink parts that are actually other types of leukocytes. And so this basically only happens in the spleen when these giant macrophages come in when there is an infection or something that they just need to check out, like something's going on, elevated amounts of toxins. And so if you see these small balls right here, the ones that those are pointing to, these are actually macrophages. And there's a few others over here that I didn't point out. You can also see in comparison to the first photo, there's a lot more of the purple area or the actual spleen area in this one compared to the second one. So it's kind of eroded a bit. So lastly, I wanted to show one more section of the spleen, and you can see that there are granular leukocytes that I circled. And these granular leukocytes come to the site of infection. It's just another type of macrophage. So you have your big macrophages on the left side right here, and then your granular leukocytes right here. And these are those white spots, not to be confused with degradation of anything. And so these types of macrophages all come together when they see that there is infection or inflammation, right? Any toxins that are building up in the spleen. And granular leukocytes are notably coming to the spleen area versus the kidney or the liver. And so those are indications of high levels of toxicity. And so that is the end of my presentation.